All right. Welcome everybody to our next round of lightning talks from our fellows. First up is Tetiana. Tetiana, whenever you're ready.
Yeah, I'm ready. So, I am Tetiana Yushkevych, and I am PhD student from Odessa Polytechnic National University at the Department of Theoretical and Experimental Nuclear Physics. Now, the title of my project is Tools for Understanding Semi-Inclusive Deep Inelastic Scattering Measurements. So, understanding the underlying structure of the nucleon in terms of partons is one of the central goals of modern nuclear physics. Partons are not directly accessible by experiment, but experimental measurements are related by factorization theorems to functions that describe partonic structure of the nucleon. One of the ways to access intrinsic motion through a process of semi-inclusive deep inelastic scattering. In this reaction, as you can see, a high-energy electron scatters off a quark within the nucleon. This quark forms a hadron in the final state, e.g. a pion, which is detected along with the scattered electron. In general, pions in this reaction are produced from the fragmentation of the struck quark is current region, remnants of the struck nucleon is target region, or from gluons radiated in the process. For data analysis, we use the affinity tool. Affinity is a new tool for experiments and phenomenological studies of nucleon structure. The tool depends not only on qualities that can be measured, but also on parameters of the underlying theory that cannot be measured. Our project connects the work on new theoretical tools with scientific software. So the main goals of our project are modifying non-perturbative parameters and recalculate affinity tool for these values. Then plot and analyze new graphs to understand whether changing the range of values for non-perturbative parameters affects the quality of the description of the experiment within one of the 4 regions shown in this figure, and to study the influence of these parameters on the affinity results and see how reliable these results are in order to guide experiments and phenomenological studies of nucleon structure. So what I already did. All the necessary literature for understanding nuclear theory in experiment has already been studied. I familiarized with Jupyter Notebook and get started with it. The limits of the parameter change were chosen, and the modifying the notebook for another experiment and kinematic region was started. In figures 3, you can see the affinity plots I have made. For these graphs, only the range of all the parameters has been changed. According to the graphs, you can see that the affinity changed markedly in this way. The next step in our project will be to change each of the non-perturbative parameters separately and evaluate the influence of each of these parameters. Thank you for your attention.
Thank you, Tetiana. Is there any questions for Tetiana? One thing I would ask too is if you have a question too, feel free to use the chat. Sometimes it helps with if there's any language barrier or anything like that. But so any questions? No? Well, thank you again, Tetiana. Very nice talk.
But I'm not. Is this okay still?
I'm sorry.
I can ask a question, but I'm not a
Sure,
student.
go ahead.
Loud. Yeah, I would just wanted to understand what kind of specialized libraries you use, because I'm rather from a field of biology, and we have like our own universe of and set of tools, which is usually unknown in other fields. So probably there is something very similar to it in physics as well. Since you mentioned that you're using Jupyter Notebook, is this Python-based libraries or what kind of?
Yeah, in Python-based libraries.
What the name?
The name, I don't know. We use the Anaconda to run the Jupyter Notebook and code on the Python.
But do you use any kind of external libraries? I don't think you implement everything from scratch.
I don't really know if we use some external libraries, because I didn't see any external libraries in these codes.
I mean, do you import anything like pandas, I don't know, NumPy, whatever libraries you use?
Maybe NumPy, and I don't know if we use one more of them.
Yeah, that's okay. I mean, then probably you're just implementing everything. Just very interesting how it is in other fields. Thank you.
Thank you for the question.
Yeah, keep in mind that many of these fellows have just started getting associated with their new work. They just started, so they may not be familiar with all of the.
No, I think this is
details.
totally okay not using anything external. It just was wondering how it is in physics, whether you need to implement lots of things or you have like the very.
No, it's a mixture. I mean, there's some things that we try to, you know, we use existing that people use existing libraries, and then there's some things that, you know, they, you know, that involve, you know, something specific to, I guess, whatever experiment or whatever you're studying. So I think that's part of it. But
okay, cool.
So, but thank you for the question. So.
All right, so my name is Bohdan, and the topic of my research in my lab is rigorous benchmarking of methods for SARS-CoV-2 lineage detection in wastewater. I do this project in Mango's lab. It's in University of Southern California. And let me tell you what it's all about. So wastewater monitoring is a different approach to monitoring trends in COVID spread and disease monitoring. It already proved itself useful in a few studies, and for example, on this graph, we can see on the x-axis, it's a graph of a history of measurements of abundance in sewer waters in San Diego. So we have a timeline and abundance percentage. And we can see that there's a green line, which means clinical samples abundance, and blue line is wastewater analysis results. And we can see that the trends are very have a certain correlation. So we can analyze wastewater to detect trends in disease spread. And on this graph, on the bottom, we can see also a study of wastewater analysis in San Diego, but here we have a timeline and lineages of SARS-CoV-2. And for alpha and delta, we can see that actually in wastewater, the lineage was detected earlier than in clinical studies. And the time it happened earlier, half months earlier. So that's great. So we have early variant detection as a success and trend identification, but we have certain issues. We in our lab work on the issue that it's difficult identifying lineages at low prevalence because in wastewater there are SARS-CoV-2 samples from the whole population of the city, basically, and it's difficult to work with low concentrations of one lineage. It's difficult to detect. Then other issues are rare lineages observed were not seen in clinical samples, and differences in sequencing protocols lead to different detection results. These are not the issues we are researching in our lab. So what plan do we have? We have a list of haplotyping tools that has been developed in the past few years, and we need to benchmark them. Basically, these tools has been developed for a slightly different purpose, and we are repurposing them for our task. To benchmark these tools, we are using a synthetic data set, which we construct by using simulation tool SimSec, which we feed with parameters we set and reference genomes for the viruses. We get an output in form of sequencing reads, a list of reads, and we test our tools with those reads. We find variants, and we evaluate the tools, and then after we're done with it, we can even get our parameters for the mixture to the wet lab, and they can generate synthetic DNA, which then goes through sequencing of DNA, and we can basically validate our benchmark in this way, our data sets, which we created synthetically. For now, we have generated a benchmarking mixture in our lab, and also I've benchmarked one tool. Thank you. And the plan is to benchmark all these tools, and also optionally there is another little list of tools.
All right. Thank you, Bohdan. Any questions?
I have a question.
Sure, go ahead.
How exactly did you generate benchmarking mixtures?
Okay, so As I said, we get reference genomes from a GSA database. It's a database which is central for COVID sequences. All the lineages are sent there, and we find parameters. And the parameters it's a big discussion of which ones to choose there. That's why it took such a long time, as from my experience. We set the number of reads, set the sequences we are going to use to make reads, and the tool makes the data set. And there are also parameters for sequencer errors. And these error error databases are specific to sequencers. There are different sequencing technologies, and as we said earlier, differences in sequencing protocol lead to different detection results. So we're focusing on a few sequencing technologies.
And another very short question, what kind of evaluation metrics are you going to use for final comparison of the results?
It's recall and precision. So it means how good the tool detects, how precisely, with what accuracy the tool detects the lineage, and how many tools of the, or how many lineages of the whole abundance of variants it detects.
Okay, thank you.
Thank you.
Any other questions? Thank you, Bohdan. Nice talk.
So the goal of the project is to develop an automatic tool to get rid of unnecessary modifiers in a statistical model specification. In what sense unnecessary is some modifiers are have a very small effect on the eventual parameters that we derive during maximum likelihood estimation procedure. And thus, it might be useful to remove them to improve the optimization time. The model is described as a product of Poissonians with some constrained terms, which are modifiers themselves. And for the rates of these Poissonian distributions, we can check whether these rates vary to a certain extent. And for example, do the following. We have the 4 normalization modifiers. We can have that the relative variation of these rates could be small, and the coefficients of the modifier could be near 1. Thus, if it is the case, we might try to remove it, just replace it with the default value of 1 and see how the optimization time speeds up, and try to see whether the optimization result doesn't shift much from the from the case of full specification with unremoved modifiers. Thus, for the how the normalization uncertainty works, it just scales the these rates, which are the histogram values, somehow provided priorly to the derivation. And for example, the nominal default histogram values are scaled by 1.4 and 0.6, and here we see that this scaling is somewhat significant. But for example, if we have an epsilon of 0.1, 0.01, we can remove modifiers which are like 0.9 or something. The next case we already tried out is somewhat more complicated because it affects this per bin rates in not in the same manner, and affects them according to the second formula in the slide in an additive way. And here we also devised a way to tell whether a modifier is negligible, and this is that we measure its relative effect maximized over all the bins. Here we, for example, have in this example picture 2 bins. If the maximum difference between these steps in an upper and lower case simultaneously is lesser than some epsilon, then we can also consider this modifier negligible. And so far, we've tried this on some small trial models and had a significant effect for some cases, like this one, where the average optimization time, which is measured like described in this slide, simply by taking the system time before and after the execution of optimizer in the library. There, on average, it's decreased by the order of 5, and not the order, simply 5 times, and the error of predicted parameters in comparison with the case where we don't throw anything out stays within the order of 10 to minus 2, 10 to minus 3. So the next thing to try out is to actually test it more extensively on more, I don't know, on larger models, on models with more complicated relationships between modifiers, and try out the most important, to try out different methods than these, like the ones which rely on taking the parameters of the model which are of interest to us, and seeing how they are affected by removing all the modifiers which are controlled by some of the other parameters. Thus, we can build a ranking scheme of all the parameters which are involved in the model specification, and try to remove all the modifiers which are controlled by parameters step by step, going from the lowest candidates from the ranking to the highest. Thus, this is the next thing to do. So far, it's been tested with the criteria described previously. Yes. Perhaps I must thank for the talk and ask if there are any questions.
Thank you, Oleksii. Any other questions?
I can ask
Okay,
again.
go ahead.
I'm just trying to understand a bit more, because it's out of my field. How general is this approach? What kind of measurements we can try to normalize with this approach? Because as I understood, you are trying to remove some kind of unwanted variations when calculating maximum likelihood?
Not really. What I'm trying to do is to reduce the time of calculating the maximum likelihood.
Okay.
Actually, these are models which are involved in particle physics experiments, and I'm not that much into that field myself, but so far these are the this is the product of statistical models which are which are responsible for each particle counter after some scattering is performed. So each Poissonian there is, as far as I understand, is responsible for some particle counter, and this histograms actually this mu zero are taken from some prior Monte Carlo simulations, I guess, as far as I understand, and to make these flexible for some particular experiments, we use additive and multiplicative we call the modifiers. These are simply the numbers which are used to
is this
to have a more to make the shape of the distribution. to make the shape of this rate histogram more flexible.
Okay, I see. Yeah, this is quite interesting because I spotted that you're using Poissonian distribution. So basically you're detecting presence or absence of some particles using like multiple simultaneous measurements. I mean, I'm trying to understand what type of data you would use, like if you want to take it from a real experiment. And funny enough, maybe it's even close to some biological studies because this is also widespread distribution for some specific tools, especially for sequencing data, because we're counting also, but not particles, but rather molecules. So that's why I was wondering how generic this approach is, whether we can potentially adapt for other types of data. This is not a question. This is rather a discussion.
Understood. To be honest, I'm rather working with this as with a black box thing. Yes, and I kind of use either an artificial data or data from data and the specifications from some published models.
Okay, cool. Yes. Thank you.
Okay, thank you. Any other questions?
Yeah. Hi, everyone. My name is Pavlo. I'm working on the project studying of Top-Quark modeling and uncertainties. Top-Quark, it's actually very cool particles because it's the most massive particle as we know right now. So if you wanna, if you've been interested in new physics, and so it's will be higher rated because due to the mass, it will be involved in very high energy, and generation of Top-Quark pairs will be generated a lot of in the background. So it's why the reason, exactly the reason why we are interested studying Top-Quark pairs. We are simulating it Top-Quark pairs by using Monte Carlo generators. There exists a lot of generators in Monte Carlos, which are simulated of different parts of simulation, from particle showering, hadronization, multiple particle interactions. Some examples of them is power HEP in Pfeffer and power HEP plus Harvick and Sharp, etc. One of the ways is analyzing it, it's by Rivet. Rivet, it's toolkit for analyzing Monte Carlo results. Rivet, it allows you to write routines, and actually has a lot of big library of already written routines, which allow you to analyze results from Monte Carlos. And the purpose of my project actually is looking through different Monte Carlo generators, and looking what is the difference between them and trying to understand which are better for simulation top-quark pairs. And the best way to do it is using Revit for analyzing that. So and here I show with one example of plots which we can plot it for analyzing different Monte Carlo generators, and this, for example, it's show up depicts single and double differential cross-section measurements of production top-quark pairs, and we can use it for seeing what difference and which are more better describes of observable data. As far as we see, for example, on this picture, that it's clear that's a red line, so which refers to power hack and PFR generator. It's describes better than blue one. So purpose of my project actually defines most best. Actually, we have a lot of this kind of generators. So main idea is to find out which actually better describes data which I have right now. Yeah, it's all.
Okay, can I ask you a question?
Yeah, absolutely.
On this slide, I can see the differential cross-section, but I don't even know for which experiment is this differential cross-section. What type of experiments you use to describe or to what type of experiments you use to find your top quarks, and for which experiment this picture of differential cross-section is?
Yeah, okay, exactly. So we have proton collisions, and we have of results of these collisions that produces a lot of top-quark pairs, and this one we produces of when this top-quarks produces. It's we are after that analyzing of channel of decades, and on this one exactly we are interesting and lepton plus jet channel, in particular. Also that means that of one top quark. So when it's collision that produces of pairs, so one top, it will be decay in electronically, and another well will be decay hadronically, and we are interested in the cross section of this channel.
Okay, thank you.
Okay, we have someone with their hand raised.
Yeah, I just wanted to add, this is Atlas data. So this is approved. The data points are approved from the Atlas collaboration public, as well as the Rivet routine.
Yeah. It's at Atlas.
So just wanted to make this clear.
Yeah.
PP collisions at 13 TV, and this is Atlas data.
Good.
Very nice.
Thank you.
I was just wondering, what are the sort of logical next steps here?
We are we are going to go through through all of the most of available different generators, and trying to pick up which are best, which are better in and why. And after that, if the time allows, to try to compare to recent data from experiments, real data.
Very nice. Okay, thank you.
Thank you very much.
All right. That's all we have for today. Thank you, everybody, for your questions, and thank you to the fellows for the presentations. And we look forward to your work at the end of your fellowship. So thank you.
Yes, definitely. Thanks, everybody.